Alright, as we have discussed earlier, essentially, ang reverse po ng catabolism ay your anabolic pathway, particularly your FA biosynthesis. O sir, anong difference? Here, class, in order to avoid your futile cycle ng ating energy use and energy generation, we should use different sets of enzymes, cofactors, and of course, these should be compartmentalized. O di ba recall, ang trend natin on our anabolic and catabolic pathways, majority of your anabolic pathways occur in your cytosol, while the catabolic pathways, majority of them occur in your mitochondria, right? That's why, di ba, si mitochondria is the powerhouse of our cell because 90 to 95%, sorry, of ATP is produced there inside your mitochondria. O class, ready na? So again, relatively shorter ang discussion natin with your FA biosynthesis because this is already the reverse reaction na lang. So at least you already know the core concept. So we also have here your membrane transport, activation, and etc. Let's have your overview. So again, for catabolic pathway, 2-carbon removal. So here naman, we will add those 2 carbon to our existing chain via the addition of your acetyl-CoA. So of course, class, we still have here your activation step. Remember ang location of your biosynthetic pathway for fatty acid for animals, cytosol. So very active in your liver adipocytes and fat tissue. Okay? On the other hand, sa plants, ayan, it's concentrated. This pathway is mainly occurring in your chloroplast. Okay? So class, ang key inputs natin here are your energy products coming from your different catabolic pathways. Okay? Particularly, we have here your acetyl-CoA, your reducing equivalent NADPH, and lastly, our energy currency, ATP. O, diba? Class, we already concluded that all anabolic pathways are energetically expensive. But class, I hope that at this point, you already observed that for anabolic pathway also, we always use your NADPH. Right? So that's the phosphorylated form of your NADH. So of course, it's very important, to use different coenzyme or your electron carrier for your anabolic pathway. Class, get ready for our biosynthesis pathway. We have your membrane transport, acetyl-CoA activation, and lastly, the chain elongation. Class, remember in our catabolic discussion for fatty acids, your activation step precedes your membrane transport. But here, you can see that membrane transport occurs first before our acetyl-CoA activation. Okay? So class, get ready with our discussion on biosynthesis. Again, this is a very short pathway lang. So please focus your attention muna on our discussion. Alright, so let's discuss first our membrane transport. O, diba? From our catabolic pathway, we know we use mainly your carnitine shuttle. O, sir, same po ba here? No, class. Again, we need to use different sets of your shuttle, your enzymes, and your coenzymes. So here, we use citrate shuttle. So it will transport or it will transfer your acetyl-CoA from the mitochondria to the cytosol. O, class, remember kasi we deliver your acetyl-CoA to the mitochondria for energy yielding pathway. So it's locked in inside your mitochondria. But if there's enough energy and thus we need to funnel your acetyl-CoA to different biosynthetic pathways, ayan, we need your citrate shuttle to transfer it back to your cytosol. Let's check. Ayan, so this is given in your manual. Here, coming from your glycolysis, remember, we will produce pyruvate. So si pyruvate can enter your mitochondria via specific shuttle as well. So class, remember, this is just your pyruvate dehydrogenase complex reaction, the conversion of pyruvate to acetyl-CoA, and this one, your acetyl-CoA to citrate conversion, is the first reaction of our TCA cycle. And that's why we can use now your citrate shuttle in order to deliver your acetyl-CoA back to our cytosol. And here, the reverse process will just occur to get back your acetyl-CoA. Sir, ano mangyayari once in the cytosol na si acetyl-CoA? Of course, ayan, your citrate will be cleaved back into acetyl-CoA for use in FA and cholesterol biosynthesis. Class, remember aside from these two biosynthetic pathway, your ketogenesis or your ketogenic pathway also uses acetyl-CoA as our initial substrate. Okay? So now let's proceed with acetyl-CoA activation. Class, diba very straightforward ang membrane transport here. Remember lang yung citrate intermediate natin in the process. Alright? Now, for the activation, class, check. Diba? Very straightforward. So here, ang activation will still use your ATP similar with our activation steps sa catabolic pathway. Remember, from fatty acid to fatty acid-CoA ang ating conversion or activation in your FA catabolism. Here, we will convert your acetyl-CoA to your malonyl-CoA via the acetyl-CoA carboxylase activity. So it will use your energy in order to incorporate a carbon chain or another or lengthen the carbon via the addition of your bicarbonate. Okay? So class, this malonyl-CoA is actually the main building block for FA synthesis. O sir, yan na po ba yung ita-transfer directly to our existing fatty acid chains FA biosynthesis? No. Okay? So class, meron tayong specific carrier protein that will be guiding our malonyl-CoA to your elongation site. And that's your acyl carrier protein or your ACP. That's why we have here your additional reaction catalyzed by malonyl-CoA ACP transferase. O class, just remember, our activated form is malonyl-CoA and your acyl carrier protein will deliver that malonyl functional group to your elongation site. Clear? Okay, so class, let's proceed with this acyl carrier protein. What is its role in our delivery on your elongation stage? Let's check. Again, just to reiterate, there are key enzymes, carrier proteins, and cofactors that are strictly required to drive and regulate our second step, the activation step efficiently. So class, recall this 3 coenzyme. So again, we have your biotin. It carries your carbon dioxide produced in the elongation step. Secondly, sa acyl carrier protein, ayan, it carries your acyl intermediates, particularly yung malonyl functional group, to your synthase complex that will catalyze the elongation step. Alright? And thirdly, sir, ano po si pantothenic acid? Class, that's just a part of our acyl carrier protein structure. Alright? And here, it's also important to note that your acetyl-CoA activation, catalyzed by your ACC or your acetyl-CoA carboxylase, serves as the primary control point of FA biosynthesis because once we converted your acetyl-CoA to malonyl-CoA, similar with catabolic pathway of fatty acids, autopilot po yan. Okay? Alright, so class, let's check the structure of our acyl carrier protein. Ayan, so class, you can see this is the head of our ACP, and in its tail, we have your B5 vitamins. Or you can see the malonyl groups must be esterified in order for it to be delivered sa elongation site of your FA synthase complex. Again, it will be esterified in your thiol functional group. That's why it's very important that we have enough concentration ng pantothenic acid in our cells. At last, this one is just your specific or detailed reaction mechanism that occurs upon activation of your acetyl-CoA to your malonyl ACP. Okay? So again, please remember these three coenzymes that we need in our activation step. Ayan. So now we are on your chain elongation process. So class, remember here, again, we only use one enzyme complex. We call it your FA or your fatty acid synthase. So all reactions, all four individual reactions occur in the specific complex. So let's check the processes and your specific reactions occurring here. So for the key process, we have for condensation, reduction, dehydration, and then another round of reduction before we proceed for another round ng elongation cycle. Let's double check these key processes. First one is a condensation process. So as you can see here, class, ayan, we will liberate your carbon dioxide in order to produce your ketoacyl intermediate. And here afterwards, your reductase, via reduction, recall, we will use NADPH. O, diba? Very clever ang cells natin. They also compartmentalize the usage of our reducing equivalents. And here, they utilize your NADPH rather than NADH. O sir, ano po ang result? Of course, formation lang yun ng double bond, your hydroxyacyl functional group. Again, class, this is the reduction process in terms of your intermediate substrate. So obviously, if this is reduced, your NADPH will be oxidized to its NADP plus form. Okay? So thirdly, we have your dehydration. Oh, very obvious naman, sir. Yes, we liberate water through your hydroxyacyl ACP dehydrase. So here, class, you can see that we eliminated your water molecule, particularly, ayan, we removed your hydroxyl group for preparation for our condensation step. Okay? So this is your reduction step. Our last step for your fatty acid synthase reaction or catalysis. So check natin. Here, we will use again another reducing equivalent in order to reduce enoyl ACP to its butyryl counterpart. O, diba? Again, another reductase will catalyze this, and this butyryl ACP will be our main elongation chain. Again, class, another round of our chain elongation will occur in order to lengthen our existing carbon chain in your fatty acid. So until it reaches your C16 or C18 fatty acid. So usually, class, in biological system, we naturally synthesize, palmitate your C16 or your even numbered saturated fatty acid. Let's check again. Ang substrate natin here is your malonyl ACP. Your 2-carbon donor with the use of our reducing equivalent, NADPH. Alright, so class, very straightforward. Just to remember these key processes and no need to memorize the individual reactions. Okay, pa? Yes, sir, ang dali eh. Okay, so class, again, this is just another visualization of our chain elongation. O class, check, naka-anchor si ACP on our fatty acid synthase complex. And this is very important in order to prevent the diffusion of our FA chain elongation intermediate until it reaches your target fatty acid. So before then, hindi niya dapat i-release yung elongation or existing fatty acid that currently elongates. Okay, so class, to summarize your fatty acid biosynthesis, let's check. TAE, di ba? Okay, class, again, TAE, transport, activation, and elongation. Again, class, for transport, we specifically use your citrate conversion, di ba? So from your acetyl-CoA na mitochondrial, we will transform it to citrate. Citrate will leave your mitochondria to your cytosol, okay? And sir, ano po ang general product? Yes, your cytosolic acetyl-CoA. And of course, this provides your main substrate for biosynthesis. But first, before siya gamitin, we need to activate it. So usually, ayan, class, again, we will just convert it to your malonyl-CoA, where malonyl ACP will be the direct malonyl donor in our elongation process. So class, remember, ang anabolic pathway, our biosynthesis occurs in your cytosol in order to build your fatty acid chain. And lastly, elongation, this is the reverse of our beta oxidation. So, diba, here, we sequentially added 2-carbon chain via your malonyl ACP. Again, similar with activation, site is in your cytosol to form long-chain fatty acids. Always remember, class, our FA synthase complex, ang naturally fatty acid that it can synthesize is your palmitate molecule. Okay? So, of course, this palmitate can still undergo post-elongation modification. So, we can introduce double bonds, your monounsaturation, and of course, this occurs in your ER, sa endoplasmic reticulum. Okay? So, that's just a supplementary discussion. End to end. Ayan, we are now on our last stretch ng biosynthetic discussion. Again, ang main regulated enzyme, si acetyl-CoA carboxylase. O class, recall, this is the enzyme that catalyzes our activation step, catalyzing the conversion of acetyl-CoA to malonyl-CoA. Alright, so class, activator niya, si citrate and si insulin. So class, again, si citrate is our shuttle. That means when we have enough citrate concentration, we can readily transport your acetyl-CoA for FA biosynthesis. On the other hand, si insulin, of course, meaning it activates glucose uptake. So technically, our cells do not need fatty acid in order to produce energy. That's why, instead, it will funnel it to your alternative biosynthetic pathway via your fatty acid biosynthesis. And for inhibitor, ayan, si palmitoyl-CoA, very familiar, that's the end product. O, sir, feedback inhibition, no? Yes. Okay? Si glucagon class, this signals that glucose is gone in our blood glucose or our blood cells. That's why we need to liberate our fatty acid for energy generation, particularly ang acetyl-CoA. That's why instead of biosynthesis, it will be fed on our citric acid cycle. So similar reasons for our AMP, energy deficient molecule. Okay, for fatty acid synthase, activator, ayan, high concentrations ng NADPH will activate your fatty acid synthase because it's its coenzyme, di ba? Your fatty acid synthase needs NADPH to complete or to successfully complete our 4 individual reactions. And of course, last again, feedback inhibition. Fatty acid is our end product here. All right, so class, to sum up our fatty acid catabolic and anabolics discussion, let's have a quick quest. Ayan, so your favorite part, di ba? So let's stimulate your brain and let's brush up your knowledge on what we have discussed earlier. Class, please pause the video first in order to guess the correct answer. Number 1, this is the site of beta oxidation. So beta oxidation is a catabolic process, sir. So that must be in your mitochondria. Very good, class. Second, this is the key enzyme in fatty acid biosynthesis. Is it carnitine acyl transferase or is it acetyl-CoA carboxylase? Sir, I remember si acyl transferase, sorry, is for the membrane transport sa catabolic pathway. Yes, class, that's good. That's very good. So here our answer is acetyl-CoA carboxylase. Okay? So class, please remember our key enzymes for your anabolic and for your catabolic fatty acid metab pathway. So thirdly, we have fatty acid activation uses blank. So again, class, ang FA activation, it occurs in your catabolic pathway. And here, activation forms fatty acyl-CoA using, ayan, very good, ATP and coenzyme A. Si NADH and FADH2, that's actually the product na po of our beta oxidation or the whole fatty acid metabolism. Alright, so second to the last class. This is the site of dietary lipid digestion. Di ba we emphasized earlier, your stomach is not the site of dietary lipid digestion. Remember, ang secretion ng ating pancreatic lipase and as well as our bile salts occur in your small intestine. That's why it is also the site of dietary lipid digestion. And last one, carnitine is required for, of course, class, we discussed this for FA transport po. For our FA biosynthesis membrane transport, we use the citrate shuttle. Class, now, we will proceed now with our very short discussion ng cholesterol biosynthesis. Let me emphasize the important information here. So again, do not worry, hindi naman siya always lumalabas sa board exam. So let's just summarize what are the processes occurring in our cholesterol biosynthesis. First one, class, again recall, our main initial substrate is acetyl-CoA. And in particular, this is a condensation reaction between your two acetyl-CoA. And remember, class, the location still is cytosolic. And for post-processing, we have your smooth endoplasmic reticulum. So for precursor, again, that's acetyl-CoA, and there, the key intermediate is what you need to know. So first one, we have your HMG-CoA. Let's locate. So this is your HMG-CoA or your hydroxymethyl glutarate-CoA class. The first two reactions are actually the reactions of your ketogenic pathway as well. But here, ayan, it will branch out via the HMG-CoA reductase to form our main intermediate of cholesterol biosynthesis, mevalonate. So again, class, remember mevalonate because this appeared on 2023 board exam on your biochem subtest. So again, mevalonate is a precursor of cholesterol similar with your squalene and acetyl-CoA. Alright, so going back for our regulated enzyme, we have your HMG-CoA reductase. So that catalyzes your HMG-CoA conversion to mevalonate. And finally, with our energy requirement, ayan, it needs ATP and NADPH because this is an anabolic pathway. For regulation, class, usually ano siya, feedback inhibition. Alright? And of course, we have your drug here or your medications that can lower cholesterol level. O sir, paano po? Your statins inhibit this key regulated enzyme, the reductase enzyme, and thus inhibiting the cholesterol biosynthesis. So kapag nag-accumulate ang cholesterol, may tendency kasi or there is a tendency to build up your fatty plaques in arteries. That's why your statins inhibit your HMG-CoA reductase and thereby lower cholesterol level. So class, remember the purpose of cholesterol, ha? Membrane component siya. For steroid hormones as well, it's a precursor, and for your bile salts as well. Okay, so class, your cholesterol is a steroid as you can see in its structure up here. Again, what you need to know here are the important intermediates. First one, si acetyl-CoA. Second, si mevalonate. And thirdly, we discussed this on your lipids, diba? Your squalene molecule in order to yield your cholesterol. Alright, so class, again, do not worry. Those are the important infos that you need to know here in cholesterol biosynthesis. And with that, ayan, we are now on our last pathway for lipid metabolism. Class, again, your ketogenesis is not always active. So this is just readily active when we are in need of alternate energy source or when we are fasting, during prolonged exercise, or during low carbs diet because we need to maintain our blood glucose concentration. Thus, our cells will find a way to generate energy without using glucose. O sir, bakit hindi na lang si fatty acid biosynthesis ang gamitin solely? Class, because here in ketogenesis, it provides a product that is efficiently packed in our tissues. Okay? So the reason there is relies on its solubility, our solubility of your ketone bodies. So let's check. Alright, so class, here again, the catabolic conversion of excess acetyl-CoA is our ketogenesis to produce your ketone bodies. And this occurs during fasting or low carb intake or generally when we have low blood glucose concentration. Alright, for our ketone bodies, we actually have three. First one is acetoacetate, second is beta hydroxybutyrate, and thirdly, we have your acetone. But did you know, class, that we only use two ketone bodies for energy generation, and those are your acetoacetate and beta hydroxybutyrate. So later on, we will discuss what is the fate of acetone. Alright, so for the location, very active si ketogenesis in our liver cells, particularly in the mitochondria. But later on, we will discover that your liver cells or your liver adipocytes do not use actually your ketone bodies as a source of energy. So main site lang siya ng synthesis. Ayan, for the reactions, we have your thiolase condensation, the synthesis of HMG-CoA, and lastly, you have your ketone bodies cleavage. Class, don't worry, no need to remember the specifics of this reaction naman, okay? And lastly, our ketogenesis is actually strictly aerobic in order to activate the enzymes participating in this pathway. Ayan, so let's check the four individual reactions that participate here in ketogenesis. First one, class, again, basic condensation of 2-acetyl-CoA to produce acetoacetyl-CoA. Diba, chain lengthening lang yan for preparation to another condensation step. Class, again, our enzyme here is thiolase, meaning we will condense your thioester molecules. Alright, second process is your condensation reaction where your acetoacetyl-CoA will be added again to your acetyl-CoA. So sir, meaning tatlong acetyl-CoA ang need natin? Yes po. So here our product is HMG-CoA or your hydroxymethyl butyryl CoA to form this compound, di ba? Sir, ang enzyme ba natin ay synthase? Yes. So class, again, here, these two reactions are similar to our first cholesterol biosynthesis pathway, di ba? So class, again, after nyan, ayan, this is the branching point of your ketogenesis and cholesterol biosynthesis. Here, your HMG-CoA through your lyase will produce your first ketone body, your acetoacetate. And this acetoacetate can further be converted to your other two ketone bodies using different sets of enzyme. Now let's see. Here, the main reaction is through oxidative decarboxylation. One is an oxidation step and one is a decarboxylation step. For your formation of acetone, we decarboxylate your acetoacetate via your acetoacetate decarboxylase, and here we liberate your carbon dioxide. And lastly, class, for the oxidation, of course, we need to use your reducing equivalent and ADH, our electron carrier, to produce our final ketone bodies, your hydroxybutyrate via the dehydrogenase reaction. Okay. Ayan, class, remember the structure of your three ketone bodies. So again, I will require you to remember the structure. Very easy to remember naman yan, di ba? Your acetone, acetoacetate, and beta hydroxybutyrate. You can see there are commonalities or similarities in their structure. And here we summarize their function and the key properties of your ketone bodies. So let's see. Again, these ketone bodies can serve as alternative fuel when we have low blood glucose concentration, or meaning our carbs are running low, di ba? So of course, aside from that, if we are poorly utilizing our carbohydrate because there are defects on our hormones, of course, our body will also resort to ketone body usage for energy production. Okay? Again, class, this is the unique property of our ketone bodies. Their solubility. That's why, aside from FA biosynthesis, we also reserve or store our energy in terms of your ketone bodies, because we can efficiently store them because they are water soluble, and hence they can be transported freely in blood without the aid or guide of lipoproteins. Ayan. For the tissue usage, again, we only use your two main ketone bodies, and this will be converted back to acetyl-CoA when we need your acetyl-CoA for energy use. So again, class, kapag nag-generate tayo ng ketone bodies and we have lack of energy, ayan, it will be converted back to your acetyl-CoA. Also, what happens to your acetone, class? We will just exhale your acetone, okay? So later, we will have a Dinkino portion for this specific ketone bodies. And lastly, ayan, again, ang liver po is the main site of our ketogenesis, but it does not solely use your ketone bodies for energy use. Again, the reason is that it lacks the enzyme beta-ketoacyl-CoA transferase. So that specific enzyme catalyzes a reaction to convert your hydroxybutyrate or your other ketone bodies back to acetyl-CoA. That's why it's actually a good thing that your liver does not use your ketone bodies. Again, it should use other energy source because it can create naman your other energy source. Again, your liver has an absence of beta-ketoacyl-CoA transferase that catalyzes the conversion of acetoacetate, our ketone body, to its acetoacetyl-CoA derivative that will be converted further to your two molecules ng acetyl-CoA. Alright, so again, class, this is very important that our liver does not use the ketone bodies as energy source. Again, in order to prevent your excess energy use, and of course, because it converted it back to acetyl-CoA, or converting it back to acetyl-CoA will use up our coenzyme A. So si CoA in the liver has many different pathways that it can participate to. Ayan. So class, again, here is the summary of our ketogenesis. For ketogenesis, again, this is the formation of your ketone bodies. And of course, the reverse of it is your ketone body utilization. So from your ketone bodies to acetyl-CoA. Very good. So for our main site, again, liver mitochondria is the site of ketogenesis. But for our ketone body utilization, that's for peripheral use of our extra hepatic tissues aside from your liver. When it occurs, of course, generally when we have low blood glucose concentration, so we need to resort to other alternative energy sources. O class, please remember, prolonged fasting, starvation, your low insulin and high glucagon, of course, they all signal that our blood glucose concentration is low. Alright, for ketone body utilization, when we have low glucose availability and high ketone bodies concentration, of course, we need to mobilize your ketone bodies for energy production. And remember our key substrates here, or our initial substrates for ketogenesis, that's your excess acetyl-CoA coming from beta oxidation, while for utilization, that's just your two ketone bodies, the acetoacetate and C beta hydroxybutyrate. And finally, for our end products, again, we have three for ketogenesis. We just add here your acetone, and for ketone bodies, we just converted it back to its initial substrate from ketogenesis, your acetyl-CoA. O diba class, again, essentially the reverse of each other din ang ketogenesis and ketone body utilization. So for tissue limitation, again, class, important thing to note here is that your liver lacks your beta-ketoacyl-CoA transferase, which catalyzes the conversion of your ketone bodies back to your acetyl-CoA. And for ketone body utilization, of course, very active in your heart, brain, and your during starvation. So class, very important that our heart and brain have unlimited supply of energy because again, continuous po kasi ang function nila. Alright, for our regulation, yan, we only have two enzymes. The HMG-CoA synthase, of course, is for the formation of ketone bodies. The HMG-CoA lyase, on the other hand, is the degradation of ketone bodies to convert it back to acetyl-CoA. And class, please remember our activators and inhibitors here in our discussion. So for our HMG-CoA synthase, when we have high concentration of acetyl-CoA, of course, that's our initial substrate. It will activate the specific enzyme. But when we have high expression of insulin, of course, meaning we need first to use your glucose for energy use other than your ketone bodies. Again, I hope that's clear. For insulin kasi, it activates or mobilizes your glucose uptake for energy production. That's why it will inhibit the production of your ketone bodies. And lastly, for lyase, activator, low blood glucose concentration. Okay? So meaning we need to synthesize or we need to use up the ketone bodies. And lastly, for inhibitors, carbs. Again, we prioritize your carbohydrates as our main energy source po. Ayan. So class, to end our discussion here, let's have a did you know segment. O, diba? Did you know that too many fats can you actually make your breath smell like nail polish remover? O ano po ba ang main component ng nail polish remover? That's one of our ketone bodies. See? Acetone, that's right. So again, your rapid beta oxidation floods the liver with acetyl-CoA. So of course, when we have excess acetyl-CoA, ketogenesis will occur to produce your three ketone bodies. And notably, your acetone. Plus, remember acetone evaporates from the lungs. They are highly volatile, and hence, we will exhale it into our breath, giving it a sweet, fruity smell like a nail polish remover. So actually, class, this is used also in clinical settings. So our acetone production can indicate diabetic ketoacidosis, meaning our body cannot use our glucose properly due to lack or defects of insulin expression. So it shifts to breaking down fat at an extreme rate of energy. So again, this can be a life-saving diagnostic clue for ketoacidosis. So again, class, si acetone pala ay merong clinical relevance. All right. So again, let's first answer the sample problem given in your manual. So I know the answer is already given, but for the sake of discussion, let's also discuss the solution for this problem. Let's read the question. When there is an excess of acetyl-CoA in the body, into which compound is it primarily converted? Is it for glucose, ketone bodies, amino acids, or glycogen? Class, diba remember in our discussion here on our fatty acid meta, particularly sa ating biosynthetic pathways, we only have three, diba? So it's either fatty acid or your cholesterol and lastly your ketone bodies. So obviously our answer here must be ketone bodies from your ketogenesis pathway. So again, we will use your excess acetyl-CoA in order to produce or to reserve your energy. So class, excess acetyl-CoA again is primarily converted to ketone bodies when energy needs are met and carbohydrates are limited. All right, so to summarize our discussion on lipid metabolism, this is your favorite slide, no? Again, we are here now on our final Lars words. Diba class? Ayan, we discussed 4 different pathways under lipid metabolism. First one, oxidation. This is just from FAs to your acetyl-CoA for energy production. That's why catabolic po siya. And recall, our trend suggests that if it's catabolic, meaning it occurs in your mitochondria. So class, again, that's a general trend that you can use. And here, remember our key step. That's the carnitine-mediated entry. That's why our key enzyme is carnitine acyl transferase. Okay, and please remember our energy products. So we had a discussion on ATP calculation for your energy products in oxidation. Okay, so for synthesis or biosynthesis of fatty acids, again, essentially the reverse of your catabolic pathway. But here, we use different sets of enzymes, coenzymes, and different location. So again, from acetyl-CoA, we will reduce or elongate your existing fatty acid chain to form our naturally occurring fatty acid, palmitate. Diba? So class, obviously, it's an anabolic process and it occurs in your cytosol because again, remember, majority of your anabolic pathways occur in your cytosol. Or remember the key enzyme for your activation to form your malonyl-CoA, that's your ACC. Okay, remember the abbreviation? ACC meaning acetyl-CoA carboxylase. And again, our natural fatty acid product, even numbered 16 carbon palmitate. Okay, for ketogenesis again, for alternative energy source, that's why it's a catabolic process. So class, again, remember it occurs in your liver mitochondria specifically. But your liver does not use these ketone bodies as source of energy. So for extrahepatic use po ang ketone bodies. So for our key enzyme, this is under your ketogenesis pathway, CHMG-CoA synthase, and remember our 3 ketone bodies. Acetoacetate, check. Hydroxybutyrate, check. And lastly, acetone. All right, for cholesterol biosynthesis, again, we discussed this is a pathway for formation of cholesterol from acetyl-CoA. Always remember class, for FA synthesis, ketogenesis, and your cholesterol biosynthesis, we use acetyl-CoA as our main initial substrate. C class, anabolic cholesterol biosynthesis, and it occurs in your cytosol, and smooth ER for post-processing. And remember our key enzyme here, CHMG-CoA reductase, catalyzing the conversion of HMG-CoA to your mevalonate, our key intermediate. Diba class? So again, I emphasize mevalonate, squalene, and your acetyl-CoA as initial intermediates for cholesterol biosynthesis. And finally class, ayan, as you can see, very busy ang mitochondria and cytosol natin for your lipid metabolism. All right? So again, class, here I would like to emphasize for our plant cells, in our chloroplast, your fatty acid biosynthesis occurs instead of it happening or occurring in your cytosol. Okay? So that's it for our discussion on lipid metabolism. All right, class. So this ends our discussion on lipid metabolism. So for now, I will cut the video first, and then on our next video, we will continue our discussion on Crash Course 8, and there we will focus on amino acid metabolism. Bye, class. Take a break. Hi class, so welcome back, and here is our part 2 of your Crash Course 8. And as I have mentioned earlier, our topic here will be focused on your amino acid metabolism. Well, let's check briefly our content for this part 2 discussion. First one, the overview. Sir, similar ba yan with your lipid metabolism? Yes po. So I will present as well here a concept map that summarizes the metabolic pathways that are relevant here in our discussion. Secondly, of course, you have your catabolic pathway, your amino acid degradation, and of course, in order to excrete our nitrogen product, we will have your urea cycle. And lastly, class, we will also discuss your biosynthetic pathway for amino acid. In particular, we will also discuss here your transamination and your de novo or from scratch biosynthesis. Class, of course, I know that you are excited to gain another set of knowledge. So without further ado, let's start our discussion here on your amino acid metabolism. Ayan, so class, here is our concept map of your amino acid metabolism. Diba? Very familiar na kayo because we also have the same concept map for your carbs and lipid metabolism. But here, class, of course, very specific for amino acid. First, breakdown or degradation. Second, the synthesis of non-essential amino acids. And thirdly, we have your nitrogen disposal via the urea cycle. Let's check each pathways here in our concept map. Starting with our dietary protein or your tisular protein, we can digest it in order to get our building blocks of your proteins, our amino acid. And of course, when we do not need energy because we have enough energy or ATP in the body or in the cells in particular, we will funnel your amino acids to your protein biosynthesis in order to get back your endogenous or your tisular protein for membrane support or structural roles. Okay, so moving forward, for our degradation of amino acids, this is a complex process that occur in stepwise steps or stepwise reactions. So of course, we will discuss transamination and deamination to get our initial nitrogen disposal product, your ammonium ion. And of course, this will be further converted to urea via your urea cycle. So class, very specific ang urea cycle in us, humans, and other organisms capable of synthesizing urea from your toxic